Hello, everybody. Thank you so much for being here today. We are so excited to be hosting this webinar today. It is the FOXG1 gene therapy update. My name is Nicole Johnson. I am the co-founder and executive director of the FOXG1 Research Foundation, and I'm also the mom of Josie, who is now 13 and a half years and has been my inspiration for starting this foundation with my co-founder, Nasha Fitter, and the team of FOXG1 parents who came together in 2017 and since then and said, we are dedicated to doing everything we possibly can to give our children a healthier life. So we have a lot to get through today, and I'm going to get right into our rundown for today. So today for our gene therapy update, first you're going to hear from my co-founder, Nasha Fitter, who is the mom of Amara, and she is going to talk about what parent-led drug development means and our roadmap to treatment. What we're doing here is something very unique for drug development, and Nasha is going to really walk you through how we got to where we are today and where we're going, and how this work that we're doing is helping to pave the way for other rare disease groups like ours to hopefully follow the same track and help accelerate treatments to patients with rare diseases. Next, our wonderful Chief Drug Development Officer, Dr. Gai Ayalon, is going to take you through the FOXG1 gene therapy program update. So Guy has a lot to share with you all today. So please make sure to watch that. By the way, today's webinar will be recorded. So if you miss anything, you will get the recording and you will be able to watch it another time. Then after Guy, you will hear from our Chief Clinical Data Officer, Ellie Brimble, who is going to talk about our clinical data strategy. So equally as important to the science and the roadmap for the gene therapy is the clinical data strategy. So Ellie will walk you through that. Next is our CFO, Ryan Phillips, who is dad to Crosby, and he's going to answer a question that we often get. And we often hear, well, what does a gene therapy actually mean to patients? So Ryan is going to talk about that. And then I am going to talk about, well, how are we going to make this possible? And after that, we will take questions and answers. We got a lot of questions in advance. Thank you so much to everybody for listening to that and submitting your questions. Please do still ask your questions in the chat. And if we don't get to them today, we will make sure to add them to our FAQ page on our gene therapy page on our website. So speaking of our website, I hope that everybody here saw that foxg1research.org got a complete makeover. We realize now is the time to highlight this program. So our new website is, we really designed it to make it easy to navigate for our three major stakeholders, which are parents and caregivers, industry and researchers, and of course, donors. So as you see right on our homepage, we talk about this first treatment ever for FOXG1 syndrome is within reach. In all these meetings we have with each other, we're really like pinching ourselves because when we started this foundation 8 years ago, we just had to first understand the biology of the FOXG1 gene. And we just said we're going to put our heads down and do everything we can. And here we are today with a brand new website and a webinar on our gene therapy update. And it is really within reach. So, and as you see here, we're making history. So something we're going to talk about a lot today is, as far as we know, we are the first rare disease foundation to drive a treatment, to drive a gene therapy through full-scale, multi-site clinical trials independently. We are sponsoring these trials, this trial ourselves, and we are on track to begin patient clinical trials next year. So this is something that, as far as we know, has not been done before, and we've had to figure out how to do it. How could we get this treatment, this promising treatment, to children ourselves? So last year, we launched a campaign for the FOXG1 gene therapy to raise the $22 million that is needed to drive this treatment through the trials to approvals. So we launched this campaign last year and we named it the Yes They Can campaign, based on the premise that our kids can live healthier lives. And there are so many FOXG1 parents here today with us, and we all know what it's like to say, oh, no, they can't do that. No, they can't do that. No, I'm sorry, my child can't talk. My child can't walk. No, my child can't eat by mouth. But what we're hoping, this work that we're doing, is that we could start saying, yes, they can. And that's what this work we do, that's what hopefully this treatment can bring us. They can live healthier lives. So we launched the Yes They Can campaign last January, and we are so thrilled to say that we are nearly halfway to our goal. Thank you, everybody. Every dollar counts. In the chat, you'll see the link to the Yes They Can campaign page. And we are halfway to the goal. So at the end of this webinar, I'm going to talk about different initiatives we have going on and how you can help us get across the finish line. So now I am going to turn it over to my wonderful co-founder, mom of Amara, and she's going to talk about how are we sponsoring these trials ourselves. And she's going to talk about what parent-led drug development really means. So take it away, Nasha.
Yeah, thank you, Nicole. This is a really exciting time. Parent-led drug development means that us as parents are really taking the helm on moving our therapy forward. We really, there's a line there. We took our children's lives into our own hands. We did not listen to no as an answer. We did not listen to, hey, there's no pharma company interested in FOXG1. We said, no, that's not going to work. Our children deserve better. And that's what we are focused on. That's what parent-led drug development really means. And it also means that we have taken the care to really create a drug that's going to work for all our children. We don't have any other incentives. We're not trying to make money. We're truly trying to create the best, safest drug to help our children. And that's what parent-led drug development means. So let's go to the next slide. So I just want to show you what drug development is for rare diseases currently. It currently costs between $150 and $180 million. It can take up to 30 years, if not longer, and that's starting from scratch. This is what the timeline looks like. And this is after I spoke to many, many biotech and pharma companies to get this, what the average timeline is. You can see a lot of time gets spent in basic science, and that is really when you're trying to understand what is this disease. And for FOXG1, when we started the foundation, there was very little understood about the condition. We didn't even have a working mouse model to understand the condition. So we were really starting from scratch and building our mouse models, building our cell lines. So many of you contributed to the donations at that time, and it feels like that was eons ago, but it actually wasn't. It was only about 7 years ago that we were literally fundraising for our first mouse model, right? And to understand the condition. So you could see that basically it's a very long timeline. It's very expensive. And if any of you haven't read the Bloomberg article that features Nicole and Josie, I would really highly recommend you do so. And you can see that even though there are amazing genetic therapies available, so many of them are left on the shelf, what we call miracle cures are left on the shelf. And that's because this timeline and the cost is just too expensive for most biotech and pharma companies to take on ultra rare diseases. They're just not fundamentally going to make enough money. And there's another graph I put on just the cooling interest that we're seeing for rare diseases from venture capital. And so all this to say that this is what we were faced with, right? When Nicole, myself, Ryan, Ellie, Guy, when we were really looking at it, this is the timeline, this is the cost, and we knew that we couldn't do this. There's no way that we could raise this type of money, and we don't have 30 years. So we have to be more innovative. And that's also the beauty of parent-led drug development. You don't have to follow the rules. You got to think innovatively. So next slide, please. So there are four things we did. We call it our four pillars of innovation. The first thing is we started our own preclinical lab. Now, you looked at the, on the previous slide, you saw that basic science timeline, that could be more than 10 years. And the reason is that there is so much inefficiency in academia right now with different academic labs, building mouse models and researching them and publishing papers. There's just a lot of time that's wasted. And most academics, their incentives are different. What they care about is publishing papers. What we care about is a drug for our kids. So look, we were very, very lucky that we have two neuroscientists that happen to be parents, Dr. Sue Kyung Lee and Dr. Jay Lee. And they have literally invested their lives now into finding a cure for FOXG1. We simply would not be where we are without them. And so what we've done, and again, thank you for so many of you that have helped us fundraise over the years, that has enabled us to fund their lab. They've been able to donate at the University of Buffalo 2 labs dedicated to FOXG1 syndrome. Like, you just do not see that for most rare diseases. We have over 20 scientists dedicated, and because of that, we have been able to create a gene therapy and actually do a successful proof of concept showing in an animal model that we see efficacy or we see positive changes from this gene therapy. That has all been done in Sue and Jay's lab, and this is why it's so important, the fundraising, and continue to fund them. The next stage is we didn't have a biotech willing to take on FOXG1. We didn't like the way most biotech companies were developing rare diseases, right? Because it was taking a long time. So we created our own biotech. Again, we were very lucky. We've been very lucky in this journey to meet Dr. Gai Ayalon, who has had a very successful career in pharma at companies like Genentech and Ultragenyx and Neumora Therapeutics, and was really empowered by what we were doing. He was excited about the idea of a patient foundation developing their own drug, taking it all the way to approval. That has never been done before by a foundation. So he's joined our team, and we've been able to create a virtual biotech that saves money. We have fractional staff, clinical operations, medical writing, regulatory affairs. We've created the framework of what we need. The third aspect is we've been really innovative. A lot of time and money gets wasted on trying to understand rare disease patients. You cannot create a drug for a population when you don't know what the progression of that disease is. And there's, as you guys might have searched, very little published on what our patients are going through, what our children are going through. So we knew that the traditional way of doing this, which costs millions of dollars, can take up to a decade, was not going to work. And so both Ellie and I are at a company called Citizen Health, which is a technology platform. And we have our natural history study on Citizen, which has really built a natural history based on our children's medical records. So thank you to all of our families that have joined Citizen. It's because of you that we have been able to have the data that we were able to take to the FDA to show that we understand the progression of this disease and pick our endpoints. And Ellie and Guy are going to talk more about that. And last is our patient support. And that is all the great work that Nicole and Caitlin and so many others are doing to educate all of you, our families, on what is FOXG1, what are we doing to create a drug, taking you along this journey with us. And so this has really been our 4 pillars of innovation, allowing us to rethink drug development. So next slide, please. All right, so this is what we have been able to do, right? This is our blueprint for how you can get to an approved therapy for an ultra rare disease drug. We've taken this 31 years down to 12 years. We've taken $180 million down to between $22 and $25 million. And this is a breakthrough. And we've been able to do it because of the preclinical lab that Jay and Sue are running, because of the digital natural history study on Citizen, and also because of some really innovative things that Guy has done with Charles River to reduce some of our toxicology and manufacturing timelines, as well as how do we think about potentially getting an accelerated approval with the FDA and European and other agencies, regulatory bodies. So that has allowed us to save a lot of costs, save a lot of time, but yet at the same time, create a drug that is safe and efficacious, which basically means that you can see a successful result. This is parent-led drug development, patient-led drug development. We are really pioneering this, and we hope to be very successful. Thank you. All right, now I am excited to introduce Guy. I think many of you have already probably seen some of the YouTube videos he's done. He is our Chief Drug Development Officer. We are where we are because of Guy's efficiency and what he's done for this foundation and how, his previous experiences, what he's learned, and how he's bringing that forward now to take this therapy to the finish line. So with that, I hand it over to you, Guy.
Thanks, Masha. Thanks so much. It's great to connect. Last time we met in a large forum was last fall in the parent conference in Florida. Some of us in person, some of us virtually. So we're now meeting maybe a little over 6 months since then, and 6 months in drug development is a blink of an eye. But a lot has happened in the last 6 months, a lot of progress, and it's great to have the opportunity to meet and give you the highlights of where we are, what we've done, and what we're looking forward to be doing in the second half of 2025. So next slide, please. So I think that the title of the next few slides is really that we are on track for beginning our patient clinical trial in 2026 with our gene therapy drug, which we termed FRF001. We have what I'll cover in the next few slides are updates on maybe four key aspects of the development program: the non-clinical and safety studies, the clinical trial planning and readiness, where are we with our manufacturing activities, and with our regulatory activities. Combined, those capture the lion's share of the scope of the work and coordination of activities that we're doing to advance ourselves into clinical trial quickly. On the timeline that you see below, which is taken from our website, so you can look at it there as well, we are sort of between those two. If you look on the right-hand side between the May 25 circle and the August 25 circle. And really what this means is that we are, if you look at the bottom arrow, we're close to finalizing our non-clinical safety studies, and we're in the midst of our clinical readiness and outcome measure activities. And if you look from that part to that point to the right, then really what we're looking at is second half of 2025, where our main focus is the authoring of our IND and its submission. An IND stands for Investigational New Drug, and an IND application is really the package that we submit to a regulatory agency, in our case, the FDA, that is the gating step for being able to initiate a clinical trial. So if everything continues to be on track as it is right now, we should be submitting our application to FDA by the end of this year and initiate our clinical trial in early 2026. So as I look forward to the next sort of coming second half of this year, it really is all focused on the application for a clinical trial and all the startup activities, operational activities that need to happen in the background, such that when our IND is cleared, we can start clinical trials efficiently. Next slide, please. So now I'm going to go over each of these sort of topics one slide at a time to just give you a little bit of additional insight into what these activities look like. So on the non-clinical side, a lot of the work was done, of course, with Sue, Jay, and the team at University at Buffalo. We've completed, since we last met, we completed 3 studies at the Center of Excellence in Buffalo. One of them was a final dose-ranging efficacy study in mouse models of FOXG1 syndrome, as well as biodistribution and safety studies. All these were done at Buffalo. through what continues to be an incredible collaboration with Sue and the team. And then with a partnership that we have with Charles River, we've completed a pilot toxicology study, and we're in the midst of our definitive toxicology study. So all these are really studies that are, their goal is to help us establish the safety and efficacy characteristics of our gene therapy. And with that, really, what is the potential, establish the potential for benefit in FOXG1 patients and the safety margins for dosing. So without conducting these studies, we really would not be able to uh, make decisions, informed decisions, and discuss them with FDA around our dose selection for our clinical trial. So this is what these activities enable us to do. Next slide, please. Clinical trial planning and readiness. So here there is a lot of work that is underway. The first bullet is one that I'll just mention here, but Ellie will cover in more detail in her slides after me. And that is initiation of a prospective natural history with focus on trial outcome measures. So many of you have participated in our past natural history study and disease concept model, which Ellie will mention, and gave, I think, a broad overview of last fall. This allowed us to design a prospective study that will carry us into the clinic. We're about to initiate a caregiver ad board. Ad board stands for advisory board. This is where we'll engage with some of the parents and the community, invite some of you to help us with input on specific aspects of the conduction of the clinical trial itself. This will in turn inform our protocol development. Protocol, we're talking about a clinical trial protocol, which is another way of saying the detailed clinical trial plan. We want to, the activities from the prospective natural history study and the caregiver ad board will help inform the finalization of the protocol itself. In other words, your voice as parents and the community's voice gets integrated into how we design our clinical trial. We have selected our clinical CRO. That is the third-party company that we'll partner with on the execution and logistical aspects of the clinical trial. And our partners that we selected are IQVIA Biotech, and we very, very much look forward to initiating our startup activities with them shortly. And then initiation of clinical trial startup activities. So this is not the start of the clinical trial itself, rather it is the activities leading up to that point. In other words, there's a lot of work that needs to happen in the background for us to be able to start a clinical trial on time, and those activities will start very soon. A lot of this work that you see on this slide is really a great teamwork with Ellie and with Mallory, which you have not met yet, and Mallory is wearing the hats of clinical operations and project management on the team. Really combine these activities will allow us to optimize our clinical trial design and launch and run our clinical trial in a timely and efficient way, such that we can generate the highest possible quality data as early as possible. With, of course, our goal is not just to start a clinical trial, but to end it hopefully successfully, such that we can move forward to an approved treatment as early as possible. Next slide, please. On the manufacturing side, so we have a fantastic collaboration with our partners at Charles River. As mentioned earlier, a lot of the work here on the manufacturing side is coordinated by Trey, who's our manufacturing expert and is pivotal for all this work. To date, we know now that we have a reliable and consistent manufacturing process yielding high quality drug product. This is not trivial. Manufacturing AAV gene therapies is an art, and getting a good process in place that generates high yields and high quality drug is not a default outcome. It's an outcome of an excellent team and a lot of hard work. We are currently using the last run that we did of manufacturing for our definitive toxicology study. And last week, we've initiated our GMP run, which is another way of saying the material, the drug product that will supply our clinical trial. So this is a significant milestone and a large undertaking. We're now generating large quantities of drug product for the purpose of a clinical trial. That's a big step and a big commitment for us. We are working closely with Charles River on all the aspects of our manufacturing-related regulatory activities and documents that we need as part of our IND filing. So, needless to say, manufacturing activities are critical for us to be able to both file an IND and start a clinical trial, and those are as well on track. Next slide. And this will be my final slide, and this is on the regulatory side. And regulatory is critical because ultimately our ability to move into the clinic and through the clinic all depends on successful regulatory interactions with the agencies. To date, we had a very successful pre-IND interaction with FDA last month. In this interaction, FDA really gave a thumbs up endorsement to all the activities that I just described to you. A thumbs up for our non-clinical and safety plans, our manufacturing plans and quality plans for clinical trials, as well as endorsement of our draft clinical trial planning. This positions us strongly to really dive into the development of the final clinical protocol. In addition to that, we have submitted recently 2 applications to FDA. One is for orphan drug designation and the other is for rare pediatric disease designation. Those are important because they allow certain benefits in terms of access and timelines when it comes to the drug development process. And those are currently under review with FDA, and we look forward to hearing back from FDA later this summer. We are on track for submitting the IND application by the end of this year. And overall, to date, had excellent interactions and feedback from FDA, which is really, I think, testimony to the hard work the entire team is putting in, and great coordination, great collaboration, and so far very successful feedback and positive feedback from the agency. I'll just add here that in addition to FDA, we are also initiating soon interactions with ex-US regulatory agencies. We can discuss this more maybe next time we meet, closer to the initiation of the clinical trial. However, this is because we are intending to any successful future drug to be accessible to as many people as possible and patients as possible worldwide. And so this is where an early dialogue with ex-US agencies, including considerations for clinical trial sites out of the US, come into play. So we'll be starting this soon. All in all, I'll summarize by saying that this is, a lot has been accomplished in the last 6 months. A lot of work is going to be done in the upcoming 6 months. And we have truly a fantastic team, and it's a privilege to be part of that team. And we look forward to meeting again closer to the initiation of the clinical trial to give more information. I'll stop here. Thank you very much.
Hi, everyone. I'm so happy to be speaking with you all today. And I'm going to share a little bit more about the clinical data work that we've been doing in the background to help increase the success of the upcoming clinical trial. Next slide, please. So when we think about developing an approved gene therapy, which is our goal, we need to do the following. First is develop an effective and safe therapy, and Guy has really provided an excellent overview of all of the work and effort that has gone into that. But number 2 is that we also need to be able to prove to the agencies that it is both effective and safe. And so how do we do that? First, we want to think about what effective actually means and how do we measure whether something is effective or not. When we look to regulatory bodies like the FDA for guidance, the FDA states that something is effective based on how it makes a person feel, function, or survive, but that's a little broad. And so we want to understand better, what does effective mean for our community specifically? What is going to be most important to demonstrate for patients, for their families, for their caregivers, really everyone involved in the care of someone with FOXG1 syndrome. So first we started by trying to characterize what would we want to measure and monitor in a clinical study or a clinical trial for FOXG1 syndrome. And so we developed two different studies that were really supported by this community. The first was our natural history study, which looked at more of an objective clinical evaluation of all of the signs and symptoms of FOXG1 syndrome and how those change over time as someone gets older. And then we paired that with a disease concept model study that looked at qualitative interviews with families to really start to articulate what is the lived experience for someone who has FOXG1 syndrome and their family. So now we're going to be moving on into the next phase in order to help support the clinical trial. So this is, not real data, but it's intended to illustrate what we might measure in a clinical trial. So we have some period of time along the bottom, and then we could imagine this vertical axis here is showing a particular measure or symptom that we want to evaluate in a trial. So what we know about FOXG1 syndrome is that it's relatively stable. We don't see a lot of changes in some of the core symptoms over time. And what we want to demonstrate with a treatment like you see here is that we might introduce a change when someone receives our gene therapy. So if we go to the next slide, the thing that we really want to be able to do and take back to the agency when we submit our data for the clinical trial is that we can really measure this difference between what do we expect to see versus what do we see now with treatment. So for the next slide, I'm going to talk a little bit about one of the studies that we're going to be running in parallel to the clinical trial. So it's not part of the clinical trial itself, but is an important companion study to it. And that's really to understand what this baseline FOXG1 course looks like for the same things that we'll measure in the clinical trial so that we can really prove to the agency definitively that our drug is effective. So in the next slide, I'll talk a little bit about what this is going to look like. So we are launching a multi-year, multi-site, prospective clinical study. That means that it's ongoing, and it's going to be very much tailored to FOXG1 syndrome and specifically the things that we're going to measure during the clinical trial. There are going to be two arms to the study because we wanted to ensure that as many people could participate as possible. The first study is going to be in-person evaluations. So if you participated, you would go into the clinic and you would have a number of assessments performed at regular intervals to look at things like developmental outcomes and sleep. And then the second arm of the study is going to be remote evaluation. So these can be done over the phone or over a video call, and we'll focus mostly on things like developmental outcomes here. So in the next slide, I talk a little bit more detail about what specifically would be included in these studies. And I do just want to call out that we're 95% certain, but there may be some changes in these as we refine the protocols over time. But in that first in-person study, the developmental assessments that we will include are going to be the Bailey, which some of you may have already had as part of your routine care. And we're also going to be using a newly developed assessment that was actually created for CDKL5 deficiency disorder, which is another closely related developmental and epileptic encephalopathy like FOXG1 syndrome. And so it's going to be a little bit more tailored to someone with the features of FOXG1 syndrome than something like the Bailey, which is broader. And then we're also going to do a physical exam and possibly a sleep study as well. And then in the second arm of the study, we'll do additional developmental assessments. So these are the remote assessments, and it will include the Vineland, the caregiver version of this newer scale, a communication measure. So something we've heard repeatedly from families is that communication is really important for us to demonstrate efficacy towards. And then also an assessment of gross motor function and hand function as well. And we'll also be including a quality of life measure, because what we want to be able to demonstrate is that not only are we able to improve the health of individuals and their overall function, but that directly impacts quality of life as well. So next slide. How can you help? First and foremost, please help us recruit and enroll in these studies faster. And the best way to do that is to confirm that you've signed up both for the registry and for Citizen. These two studies are how you let us know that you're interested in research and you're interested in our gene therapy trial. And so this is how we know who to invite to enroll in these studies. And it will likely be a requirement for the clinical trial that you're enrolled in both of these as well, just to make sure that we have as much data as possible. Next, if you're interested in enrolling in one of these prospective clinical studies that I've just described, please do so when they open. And I will just say that every time point is so important in these studies. So if you do want to participate, the best outcome for us is if you're able to join for the full study and to participate at each time point as well. So that's going to be my last slide for today, but you'll certainly be hearing more from me as these studies start up, and I hope many of you will consider joining them.
Okay, great. Thanks, Ellie. Super excited to be with everyone here today. So one thing that I thought would be informative for us as a community to walk through is what we're seeing in AAV9 gene replacement therapies in other similar disorders to FOXG1 syndrome. To kind of answer the question as to why we feel that AAV9 gene replacement therapy is a good candidate for FOXG1 syndrome. And so these that you see on this grid on this slide are similar disorders. These are monogenic neurodevelopmental disorders that share similar symptoms to FOXG1 syndrome. And so on the left-hand side, you'll see the disorder. We have spinal muscular atrophy, Rett, and GAN. And in parentheses under them are AAV9 gene therapy drugs that have been developed that have either gone through or are in clinical trials that have made disclosures or reported outcomes of treated patients. And so along the top, you'll see the symptom domains that are shared with FOXG1 syndrome. So we have motor, we have communication and social, and then daily function and health. And so if we look back in history at this drug modality used in similar disorders, to try to look forward as to what we may see in FOXG1 syndrome, and we look at motor, for example, patients or children that are born with SMA, type 1, for example, unfortunately, the condition is lethal and they don't live to see their sixth or seventh birthday. And this drug not only saves their lives, but they also obtain motor milestones like the ability to sit, stand, walk, improved head and limb control. These are milestones that they would never reach. In the Rett trials that are being conducted both by Taysha and Neurogene, we're seeing children and adults regain skills that they had lost, like walking, sitting, functional hand use. And we're also seeing children and adults gain functional skills that they had not prior had. So again, walking, fine motor skills that were not expected at their age based on the natural history of how Rett plays out. And then if we move to communication and social, across this domain in all categories, we see increased eye focus and control, increased engagement, increased vocal sounds and social interest, increased connections, and general responsiveness of these patients post-treatment. And then in the daily function and health domain there, We see in SMA, these children unfortunately need breathing support and often feeding tubes. Less of those are needed. These children are more stable. They don't need that support. In Rett, we're seeing improved sleep outcomes. We're seeing fewer seizures, fewer seizure medications, improved swallowing, improved constipation, greater stability in GAN. And so these are really life-changing outcomes, as you could all imagine, for our children and for our families. And then a couple of other notes are that in the SMA row, for example, this is a single dose given that has long-lasting benefits. These children, I think the drug was approved in 2016, and these children, it's not a transitory effect. They're not regressing back to baseline. These are long-lasting outcomes via single administration. And then some other notes is in the Rett trials, these are in trials now and have disclosed interim analysis data just weeks after treatment. They saw some of these significant improvements in both children and adults. So again, we're cautiously optimistic based on history and how this has played out for other similar disorders. Which, if you could go to the next slide, leads to why we think FOXG1 is a good candidate for AAV9 gene replacement. So one is it's also a monogenic neurodevelopmental disorder, like the other disorders that we just viewed, in which they have demonstrated that the brain is plastic and that it can be rescued postnatally with treatment to regain functions that had either been lost or never developed. So we're optimistic that this fits in that same category. Number two is we see neurodevelopmental disorders with shared symptoms, as viewed in the last screen, have meaningful improvements, increasing quality of life, increasing the ability for our children to have more full, happy lives. And then three, and Guy talked to some of this, is we have some very exciting preclinical data that's coming out of the University of Buffalo and the Lee Lab, where we see restoration of both brain structure abnormalities and cellular abnormalities in those brains postnatally that lead to improvements in those lives. And mouse brains are not human brains. We understand that, but we're very excited by the preclinical data that has come out from the Lee Lab, which has just been a fantastic partnership for us. So these are the three top reasons why we believe that FOXG1 is a good candidate for AAV9 gene replacement, and we're very excited to hopefully realize that future for all of our children. And I'm going to turn it over to Nicole, who can help talk about how we can make this happen.
Thank you, Ryan. That was great. So as everybody's seen here today, we have the team in place. We are moving swiftly along this gene therapy roadmap. We feel very strong that this is the most promising treatment for FOXG1 patients right now. And we've, from when we started this foundation in 2017, we have dedicated our lives to doing this. We've dedicated our lives for all the children across the world. We made a pact that this is not just for our children. This is for all the children in the world with FOXG1 syndrome today and in the future. And I always say this is for children we won't even know one day. So what we're doing here is, really changing the future and changing the experience for families like ours. We know how hard this experience is. And if every bit of work we do can help make life easier and better for our children and just give them more of what they deserve, a life without suffering. Most people here in this webinar today are parents. So you've seen us work, live, breathe this. And we've gotten to this point where we have a real treatment for FOXG1 syndrome. Close. Guy showed you we are on track to begin clinical trials next year. And as Nasha showed you, we found a way to do it and to control our own destiny and not risk, our treatment being put on the shelf. So we have innovated at every step of the way and just worked smart and efficiently with the goal of all of our children. So we figured out how to go from something that could cost $180 million, took it down to $22 million, which is a fraction, but all the money that we have raised has all been our own efforts. We have done this all ourselves. Everyone here, all the fundraising teams that you've all done, all the events that everyone has done, every single dollar has come in from friends and family and going out there to companies, sponsorships, everything. So we have raised 10 towards our $22 million goal, but we still have 12 to go. So how are we going to get there? And this is next slide. This is when we need your help more than ever. This is a community effort. We are parents that started this foundation, but we have to all do this together because, if everybody helps, we will get there. And we need all hands on deck. We cannot do this alone. So what I want to say is ask yourself, what can I do today that can help get us closer to our goal? And there are many, many ways you can help, but I want to point out one incredible man and one incredible initiative that is happening this summer. So many of you have heard of Tom Horton, who joined our foundation a few years ago, and he is chairing our philanthropy board. He is the grandfather to Gianna, and Tom is one of the most celebrated businessmen in the United States. He ran the world's biggest airline at the time. He's the former CEO of American Airlines. And when Gianna was diagnosed and he looked at what the FOXG1 Research Foundation is doing, he actually did once he joined us, he did say, he's like, you are the most efficient group that I've ever worked with, which actually that scared me. I said, you ran an airline. So Tom joined us and has really brought in his network, has dedicated his life, his family to helping us drive treatments to children. And we have Tom to thank for a lot of the fundraising that we've done. Every single dollar helps. And so we've got to this point where we have to keep the momentum going, but we need everybody to help. This is not on Tom alone. And what Tom is doing this summer is he is embarking on a very special walk. It's a pilgrimage of prayer. It's called the El Camino. So Tom is walking the way. It's a very famous walk across France and Spain. And he is going to walk 500 miles for FOXG1 children. And he's doing this to raise awareness, to help get sponsors for his walk. Every step is to help get us across this finish line. So now we need to all help Tom. So different ways we can help power Tom's walk is through sponsorships. We just got our first sponsorship. Blockchain.com has come in with a sponsorship towards Tom's walk. So if anyone knows any companies that would like to align with this mission, this very profound event of Tom walking 500 miles and praying for children along the way, we're looking for corporate sponsors. Another thing you could do to help power Tom's walk is dedicate a mile to a child. So Tom will have a list of FOXG1 children and he will pray for them along the way. This is a spiritual journey. And so for every mile that is dedicated to a child, Tom will have that child's name in a list. And he will be praying for all of our children, and doing every step will help get us closer. So that is one very exciting thing that hopefully everybody can help us power Tom's walk. And there are many other ways that we can all come together and help. So obviously, we can always donate. Every single dollar helps. I did a small event last night. It was, a Monday night event. It helps. Everyone helps. And we have many ways you can donate. Crypto. Got any Bitcoin? Stock, Wire, DAF, DAFs. So in the chat, there's links. Donations are always helpful. Help fundraise. So we've created the Yes They Can campaign page on our website, as an easy tool that you can share with anyone. So it tells the story. It's all in one compact place that very transparent of what our goal is for. With our new website came a new fundraising teams. So everyone could set up your own fundraising page. It's your own URL and write your story, share it with your network. And if we all create our own team, we could get there faster. If we all host an event, across the world. We are here to help you host events. We've put together a team of FOXG1 parent volunteers, calling them FOXG1 event navigators, that are here to help you make your event a success. So many people have been doing events in our sister foundations in other countries, Spain, France, all around Europe, Australia. Thank you so much for all your incredible work. We love seeing these photos of all these different initiatives that are happening around the globe. I look at it like we have this one giant boulder. I said this at the conference, with hands from across the world pushing this boulder uphill. So if everybody is doing an event, just like I did a small thing last night, then we're going to get to our goal faster. And something else I want to share with you is that our leadership team is on a philanthropic roadshow. So we are meeting with prospective donors all over the world by Zoom, in person, to share our story, because there are so many philanthropists out there that are looking to make a big impact. So if we have the opportunity to share the story and the impact of their support, then it's very likely they may want to support us. So if you know anyone, any philanthropist that you think would be aligned with our mission, please introduce us so that we can set up a meeting and share our story with them. And the one thing that we share with them, well, it's three things, is the impact of support. So next slide. The impact of support are three very important things. First of all, it's near term. We are beginning clinical trials next year, and I want to make clear that the goal is not the trials. The goal is an approved drug. But by beginning trials next year, we are that much closer to having a treatment for FOXG1 syndrome. So the impact of support is near term. It's also potentially life-changing. For us as parents, the smallest things are, what might seem small to somebody else, but if our child has some words, if our child can stand up, some of the things that Ryan talked about that we are seeing with gene therapy, with other diseases, are life-changing. So the impact is potentially life-changing for our children and for our families. And the third thing, which Nasha really pointed out, is that this is far-reaching. There are 400 million people in the world living with a rare disease. As a parent-led organization, we have found a way to take that timeline from $180 million in 31 years down to 12 years, which we're already 8 in, and $22 million. So we are paving the way to accelerate treatments for rare diseases at large. We're accelerating rare disease drug development. So taking a bet on us and helping us drive this treatment to the finish line really has an impact beyond measure. So with that, we will begin questions. But before I do, I just want to say thank you so much to everybody for every ounce of support, for all the encouragement along the way. And this is truly a community effort, and we are all in this together. So thank you so much. So now I am going to get into the questions, which we got a lot in advance. And I will say, going forward, the place to ask questions will be on the form on the FAQ page. We will continuously be updating that page, but as you can imagine, we are getting questions in every medium possible, on Facebook chat, everywhere, but the right place is on that form so that we could systematically answer them. So, okay, I'm looking at the questions here. This is a great one, very timely. And I am going to give you this question, Ryan, because we talk about this at length. Will the federal cuts to medical research impact the funding for FOXG1 gene therapy? Great question. Ryan, I think you are best suited to answer that based on our recent discussions.
Yeah, thanks. Yeah, really good question. So, the short answer is directly no. We have not to date received financial support from the federal government in which cuts to budgets are going to impact us. But there are indirect implications, obviously, of the NIH cutting budgets and funding to academic centers. So, one thing that we are very fortunate to have such a strong community and have been to date successful in fundraising is we've been able to shield ourselves from both federal budget cuts as well as pharma or biotech cyclical downturns because we are driving this therapy forward ourselves. And so through community support and the ability to continue to be in the driver's seat and bring this trial forward as the sponsor, we have walled ourselves off from some of these impacts. So to date, no, but I will say that we need to continue to raise funds to remain in that position so that we are not impacted by these types of downturns. And we can continue to support like the University of Buffalo lab, for example, who without our support would be significantly impacted by budget cuts from the NIH, that would impact our drug development processes.
Thank you, Ryan. Great. Okay, so I'm going to ask the questions in buckets now. So these are some questions about the clinical trial process. So Guy, I'm going to turn to you to answer these questions. So the first one, for those enrolled in the gene therapy clinical trial, will receiving this therapy prevent them from being eligible for future treatments? It's a good question.
Yeah, it is a good question. And I think the best answer we can give is at this point in time is not necessarily. It will really depend on the specific treatment. As far as we're aware, at this point in time, there are no additional other kinds of treatments that are close to being ready to enter clinical trials. So it's a little hard to say specifically for one treatment or the other. That said, I think that there are different modalities in treatments, and so participation in this trial doesn't necessarily preclude, but that will be really case by case dependent.
Great. So also about the trial process, one mom asked, how is the therapy delivered? So how does it happen?
Yeah. So the delivery of the treatment is through what we call ICV injection or ICV administration. ICV stands for intracerebroventricular. This is a fancy way of simply saying that the treatment is injected directly into the brain, into a specific space within the brain where the cerebrospinal fluid is being made, and that allows for distribution of the drug throughout the brain. The reason there are different ways of administering gene therapies. We see them with different ways that different companies are approaching. Some are like we're planning direct injection into the brain. Others can do it through an intravenous administration. The reason that we're going with this route to administer the drug is because that way we maximize the amount of drug that reaches the organ that we are targeting, which is the brain. We want the brain to have as much, drug exposure as possible, of course, within the scope of safety. But this is the organ that needs the supplementation of the FOXG1 gene. At the same time, this approach of administration also reduces the exposure of the rest of the body to the drug. This is an advantage because we have more where we need it and less where we don't. It is important because we have received different questions and understandable questions around safety. We know that there have been reports of safety events with other drugs, some of them approved, some of them in clinical trials, and safety is front and center on our minds. We are paying a lot of attention to that. As much as we all strive for efficacy and treatment effect and benefit, first and foremost, we look to safety, right? And this is a key consideration. And so it is, when we look at the safety events that have been reported for other treatments, they have all been associated with high concentrations of the drug in the periphery. When we say periphery, we mean basically the body outside of the brain. The brain is where we're focusing. The periphery is everything else. And the safety events that we've seen have to do with immune responses, as well as just high drug concentrations in other organs of the body. By administering directly into the brain, not only we maximize how much the brain is receiving, we're reducing how much the rest of the body is receiving. And as we consider our doses, which we've also discussed with FDA, those take into account the threshold of drug concentrations that led to safety events in other cases, and we're ensuring that we are going to stay way below that threshold. So all this is to say, our route of administration is directly into the brain to maximize efficacy, to minimize risk of safety. And that's the path that we're taking, as well as, and we see this with other sponsors as well that are now moving more and more toward that route of administration for the same reasons.
Thank you. Yeah, you answered a lot of other questions with that as well, because safety and side effects, these are big questions that we get often.
Of
But
course.
back to the process of the trial, and I'm going to ask these two together. How many doses? So a gene therapy, how many doses, how often, and where does it happen? So what would a parent expect to
Yep.
bring their child to a hospital? Is it a one-time deal? How does it work?
Yeah, so the treatment itself is a one-time treatment. It is what sometimes is called one and done. And as Ryan described in, when he talked about the studies, Zolgensma for SMA, those are also one dose, one administration, and so far we see long-lasting effects over several years. This is the approach that we're taking with our treatment. In terms of the process itself, in terms of where does the treatment take place. So by injecting the treatment directly into the brain, this naturally requires a surgical procedure. So this is done in a hospital setting. So the dosing itself will happen in a medical center, in a hospital, and it will involve a certain period of time of hospitalization, just staying in the hospital or close to the hospital, and this is part of our prioritization of safety. We want after administration to be able to monitor very closely to make sure that there are no anything that needs to be responded to from a safety point of view. After that, it's home and back to life with periodic. This has to do more with the protocol that will be developed later this summer. But for the duration of the trial, only the beginning is the dosing in the medical center. After that, yeah, the assessments and follow-ups, some of them are going to be remote from home, occasionally maybe a visit to a doctor's office. More details on that we can provide probably in our webinar that'll happen closer to the clinical trial itself.
Right. So a lot of the questions that were asked, we won't be able to answer until that next webinar when the protocol's been designed, and we're much closer to the trial. We do get a lot of questions about trial design. So I'm going to read a bunch of them at once for you, Guy. And of course, what we can answer now and what we cannot answer now, we will and we won't. So a lot of people are asking about trial design and criteria, of course. So are there exclusion criteria, existing medical conditions that might prevent participation? Is it necessary for the patient to be a US citizen? What are the ages of the trial? Really, we get a slew of trial design and participation questions.
Yeah. So everything related to inclusion, exclusion, specifics of, will my child be able to enroll in the trial, etc. All these we're going to provide more information next time we meet closer to the clinical trial. It has to do with the clinical trial protocol itself, which we need to finalize. It needs to be, again, vetted and approved by FDA. So I know that those questions are important, and we will address them as early as we can. It's just that we're not there yet. With regard to the question around U.S. citizenship, the answer is no. It is not required to be a U.S. citizen in order to be able to participate. I mentioned in sort of my last sentence of the last slide was around us engaging with ex-U.S. agencies as well. This is where, again, we'll provide more information in the future, but we are assessing, opening of clinical sites for the clinical trials outside of the U.S. as well. And so, first of all, there will be some opportunities to be participating outside of the US. That said, ex-US, whoever lives outside the US, can come to the US as part of the clinical trial. Here, it is important, maybe Nasha, you can chime in here. We did mention earlier that it will be important and actually essential for whoever wants to participate in the trial to be in the registry and part of the natural history. And here, Nasha, if you can maybe elaborate a little bit about ex-US participation in the citizen study, how that can happen.
Yeah, thank you. Yep. It's fine. Thank you. Yeah, there's a little bit of confusion there. So first, everyone should be in our registry on Benufit. Brianna's posted the link in this chat, and it's on our website. If you don't do that, we don't know you exist, honestly, and we need to confirm that your child actually has FOXG1 syndrome. So coming there is the first step. And then, yes, we've created Citizens so we can get a deeper understanding of the progression of the disease. Any patient anywhere in the world can join Citizen. In the United States, we will go and collect all of your medical records, and that's a benefit to you. If you're outside of the United States, unfortunately, at this time, you will have to upload your own medical records. That's not a requirement. The only thing we do ask is if you could upload your genetic testing report. Again, we need to confirm that your child does in fact have FOXG1 syndrome for you to be eligible to join a clinical trial. So though that's a critical thing. But again, just to reiterate, because there's a lot of confusion, anyone can join Citizen. We just won't be able to collect your medical records if you are not located in the United States, and we need you to upload at the very least your genetic testing report. So hopefully that's clear. And this is really critical as a first step.
Yeah, and your genetics report has to be in your
registry. Nicole, you're on mute.
No, it wasn't. Do you hear me? So your genetics report also has to be in your registry. So the first step for every parent, if you are interested in being in any study, is to have your registry profile done with your genetics report uploaded. If you have any issues with your genetics report, one parent wrote in the chat, reach out to us. You can email us. Peggy at foxg1research.org is helping with our clinical development data, and we will work with you on it. But so your registry with your genetics report, have a citizen account no matter where you are in the world. And if you are in the US, absolutely, they will access your medical records. So they're very simple things to do, and it really leads to the advancement for all of FOXG1. Okay, great. Thank you for those answers about trial design. So the next one, I think Ellie and Guy, you could speak to this. And Ellie, you really did lay this out in your presentation, but what outcomes are you hoping to achieve with this therapy? We saw outcomes for other diseases that Ryan pointed out. What are we hoping to achieve with our FOXG1 gene therapy?
Yeah, that's a great question. And I think part of what is both exciting and scary and many feelings about what we're doing is that we're the first ones to do it. So there's never been a clinical trial or a therapeutic developed specifically for FOXG1 syndrome before that targets the cause of FOXG1 syndrome specifically. So a lot of the work that we've done both in the preclinical setting that Guy has described and in some of the clinical work that I've described, is to arm ourselves with as much information as possible to try to be thoughtful and make thoughtful predictions about what we could expect to see. So we'll be monitoring in the clinical trial symptoms that we know are both common, impact quality of life, and have been shared from families as being important to the patient's health and the patient's and families' quality of life as well. And that's what we'll be measuring to see whether we can observe differences there or not. Of course, we will learn as we go, and just based on our mission and this being community driven, we will certainly be looking for and creating avenues to share what we're learning back with the community as well. Guy, I'm not sure if there's anything you wanted to add there, but please feel free.
Yeah, no, nothing to add. Thanks, Ellie.
Good. That gives us a few minutes for a question that we get a lot. A lot of parents want to know after the clinical trials, what next? So, and that's really important because the trials is not the goal. Yes, we need to raise the money so that we can get through the trials, but the goal is an approved drug around the world. So parents want to know, what does that look like? Once we get through the trials, how long will it take to get an approved drug? A lot of parents are asking, I spoke to a dad the other day, should I be saving up all the money because I'm going to need it for the drug once it's approved? Well, we actually have to raise the money so that we can get there. But so the questions about after the trial, if the therapy is successful, how soon will it be made available to other patients? Will it be available outside the US? How will parents access it once it's approved? So Guy, we talk about this a lot.
Yeah, we certainly do. And our goal was, is, and will continue to be to get to an approved drug as early as safely possible, and to make it available to the broadest possible patient community of FOXG1 syndrome worldwide. This is the vision, and our entire program is designed based on that and geared to that. Now, there are things that are, like everything else in life, there are things that are in our control and things that are not. So everything that is in our control, we're doing in order to enable that vision every day, right? That's our daily work, right? There are things that are beyond us. We can't control the data that will come out. We can't control the regulatory agencies that we're interacting with. We can't control the market in terms of, payers and market forces that dictate drug pricing. So I think that what we can say at this point in time is that we design our strategy in order to enable the fastest possible path for approval, because that is what we can mostly control. And then together, and this tightly dovetails with what Ellie discussed, part of our goal of generating a robust prospective study that will go in parallel and in partnership, if you will, with the clinical trial, is such that when we come to the end of the study, we can demonstrate to whether it's FDA or any other regulatory agency the robustness of the data. We have the ability to demonstrate that this drug works if it works, right? We need the data. And those are the things that we can do in order to to shorten as much as possible, together with other creative things that we're doing in order to accelerate timelines. However, once it's in the hands of a regulator, how long it'll take for them to approve depends on their bandwidth, their workload, things that we don't really control. So, what we see in general, that it can take sometimes a year since the FDA, from the time that it gets a package till the time that it approves it. And then from the time that it's approved, how long before it gets to market. And all those things are down the road. They're on our minds. We will, just as now we are dealing with how we make efficiencies to run faster today, tomorrow we'll deal with the questions of tomorrow in the same manner. I know that it's not necessarily satisfying because we'd love to have all the answers to all the questions, but it just, I wish we could. But what we can, we're addressing, what not, we'll have to see down the road. We will do the best we can to also influence pricing as a patient advocacy group. If we were a company, it's harder to do because then our revenue would depend on that. But as a patient advocacy group and a nonprofit, I think that we're better positioned to influence pricing, but to what degree and how much it's going to cost, it's just too early to say.
Thanks, Guy. Yeah, well, that is about time here. We will reach out to everybody who's here with a recap of everything we talked about, with all the links. We can't ask for your help enough. You know, we've gotten this far. We really need help to take this to the finish line and get a treatment for all of our kids. And we will continue to do this work. We will continue to live, eat, breathe FOXG1 syndrome. We promise you that. So please join us. Please, this is what keeps me up at night. We have 12 million more to raise, and we can do it if we all do it together. Share the links. I saw somebody said they want to share Tom's walk with their church. That is an amazing idea. Getting local sponsors from your towns, wherever you live, they will support you. They know your families, they know your child. So reach out to us. There's a volunteer page on our website. Join us. If you want to host an event, please do. That would be great. Start your fundraising team. We see when somebody else starts a team. We see when the donations get in. We are grateful for every ounce of support. As you can see, I've been blessed to work with this incredible team. It's just this work really is a joy. And knowing what we're doing here is really incredible. And we're just so grateful for every minute, ounce, dollar of support, and we will communicate as best as we can. And you're on this journey with us. That's what's most important. This is about all of us, and we are going to communicate with you the best that we can, and we're on this journey together. So thank you for being here, and we will send you the recording of this video. Have a great day.
Thank you,
Thanks,
guys.
everyone.
Thank you. Thanks, everybody here.